Thanks a lot. Thanks a lot for the invitation and for this opportunity. It's very nice. Um, and yeah, I think a scientific talk, I think, is a little bit of a big word. This project really started as a weekend project. I mean, it took a much bigger shape and size on the big weekend project. But initially, when ChatGPT came about, I got really excited about it, like all of us. And at some point, I kind of thought, oh, I should probably try to have a project, try to do a project at the intersection of NAPARI, which is this n-dimensional image viewer in Python that I started with my friend Juan Inez Iglesias, and that now has a huge team of developer all over the world, that are working on it to improve it. So I was interested in basically, seeing what can be done at the intersection of this new AI, this new way of basically having agents that can, reason and act in our world, and connect that with Napari and see what could happen. So a little bit of background about us. I think that's useful. So we are at the Chancellor Gruber BioHub in San Francisco. This is a kind of a hub between the three universities there, Berkeley, UC7, and Stanford. We have investigators from these universities that are funded by us, but we also have an intramural research program that I'm part of, and that's how I started at the BioHub. Now I'm leading one of the programs, but initially I was a group leader, and I still have my research team, of course. And what we do is, what we're trying to do is really, our dream is to integrate developmental, to build an integrated developmental cell atlas that connects cell dynamics, lineages, identity, and state. And to do this, we use a vast array of technologies, from imaging, genomics, bioengineering, um, um, computer science, and AI, and also genetic engineering. And we use all of that, and we try to establish this map of cell dynamics, cell lineage, fate, identity, and state in zebrafish, which is an ideal model organism because it's very transparent. We can access the embryos early on, image them, and do other things, other measurements coupled with imaging. So one quote from Siedenbrenner that I really like is, "Progress in science depends on new techniques, new discoveries, and new ideas," probably in that order. And that quote is, I think, important for me because the order in which these things are stated, I think, is key. If you do not have new techniques to see better, see further, push the horizon of what's observable, you won't be able to necessarily make new discoveries, because simply some things are, not everything, but some things are hiding beyond the horizon of observability. So the key here is to, for us at least, our approach to doing science is to push the limits of what's observable, and then work back from there, work back from the biology that we're trying to uncover, and basically do our science in this way. So what that means for us is we have to build advanced instruments. We build advanced light sheet microscopes. We then produce, terabytes and soon petabytes of data, our latest generation microscopes. Each image, and I think Ilan could intervene there because I'm not sure I remember exactly the size, but it's in the order of half or a third of a terabyte per single image, 3D image, because the raw data is made of a lot of images. And so
half a terabyte.
half a terabyte, which I still can't
Yes.
I still can't believe that's true. It's like, that sounds like a whole data set, a whole time-lapse data set, but this is just a single time point of a data set. And so our image processing, image analysis, problems you can imagine are tremendous there because of the completely crazy scale of the data we're generating. And Ilan, actually, with whom I'm tag teaming today, is a software engineer, a very talented, computer scientist. He is actually at the core of he's basically doing the work of massaging this gigantic amounts of data out of our microscopes. I can tell you, he's done very well, but it's also he was telling me yesterday he's very exhausted because the enormity of the task is massive. And so we process these images and then we produce data sets that we then analyze with AI. We do cell tracking, we do cell segmentation, and then cell tracking and other things after that. And we don't just build these technologies for building them. We build that, we establish these technologies both at the computational and optical level to be able to do a developmental biology of whole zebrafish embryos. We image them. We do also omics. We use microscopes that you can buy, which are sequencers that will just very fancy microscopes. So we use all these technologies that we either design, develop, or buy, and then we throw all of this fanciness at the question of how embryos develop, how embryos are built, how cells self-organize to build embryos, and that's really the mystery we're trying to address. And so we've done a lot of work on the technology level. That's what you're seeing on the two left panels. Going from new microscopes, new versions of our microscopes. And Napari, as I mentioned, a tool that's taken a life of its own, with under the guidance of Juan, who is still very much involved, and many others in the team. We do a lot of terascale image processing, embryo scale cell tracking, and what I will talk to you guys about today is how we use foundational models, LLMs to do image processing and image analysis. And we also have, of course, as I mentioned, we have already, our first kind of flagship paper came out last year in Cell, ZebraHub, where we combined imaging and sequencing to understand zebrafish embryonic development. And we have more projects on the way, especially before the end of the year. So as I said, it's all a means of then, and one of our first deliverables there on the biology was this project that was led by Merlin Nang in my team. Now he's in Paris, starting his lab, and he's going to do amazing things from what I'm hearing. But in my team, what he helped me do is establish zebrafish and build up this resource that combines light sheet imaging and single cell sequencing. I will go a little further and faster now, because I think this is not the purpose of this presentation, but I just wanted to give you an idea of why we did this AI plus bioimage analysis. And the reason is that we really need to push the limits of what's possible in terms of processing images. And so, of course, using AI in all possible forms was key to do that. So as you have noticed, for the past 2 years or so, time is flying, it's a little bit more than that now. We are past, actually, these are slides I already have for some time, and we are definitely past the threshold of the AI revolution. We are squarely in the AI revolution. And the question is what will come next? And I think we are all realizing that things are moving extremely fast. And so what we'll present to you is Omega. But what's going to be interesting is that Ilan is going to show you the next thing. And it's moving so fast that there was no point for me to actually do a demo of Omega, because we already have something better. So where this all came from was, early on when ChatGPT came about, I was asked by Rita Strack, at Nature Methods, to write a little piece about the future of bioimaging. At first, I was a little bit, unsure what I wanted to talk about, and I was very busy. So I told Rita, I will try to write something. I'm a bit busy right now. And she told me something along the lines of, oh, don't worry about it. If you're not ready, we can move on without you. And that had the effect of motivating me to actually figure out something. And I was very grateful to Rita for that little nudge, because then at that very same moment, ChatGPT was in the air, and I thought, okay, let's write something about the intersection of large language models, this new kind of AI that seems to emulate human intelligence, and bioimage analysis. And this led to a piece where I imagined a future where we would basically do bioimage analysis through a conversation with the machine. You would provide the images, you would provide the tasks, you would say, I want to segment this image, I want to count the number of cells in this image. And the machine would start working on this, and you would have a dialogue with it. You would look at the output and give it feedback, or change, give it more data. Maybe it would give you back an image. It would show you an image back and say, is this what you wanted? Is this correct? And through a whole conversation that might last an afternoon, maybe what we would normally need, perhaps, a whole PhD of a bioimage analyst, maybe we could do the same thing in an afternoon. That would be required, that in the past would require a whole 3 years of work. So of course, I imagine this would happen in 10 years from now. But of course, we are at an inflection point in history, and I think this is of course a revolution of historical significance. We're at an inflection point where things are moving so fast. Currently, we're still in an exponential phase, so basically our intuition cannot grasp at how fast things are going, because they're always going faster than we ever can imagine. And so we are at a point where what I thought was possible in 10 years is practically possible today. And of course, if it's not possible today, these things are never going to be, this is the dumbest they're ever going to be today, right? So just wait another year, and they're already like, orders of magnitude better in different, over different dimensions. So at that time, I was not going to wait, years for this to happen. I was wondering, how much is possible today, and I was thinking, well, can I make a prototype that already like puts all the pieces together, and perhaps as the pieces get better over time, this would mature and maybe become what I thought could be possible in the piece. So I started this project Omega, which is a ChatGPT based NAPARI aware conversational agent that specializes in image processing and image analysis. So NAPARI is this very cool project, that is essentially a Python viewer for n-dimensional images and meditation.
Yeah.
Hello? And so basically, NAPARI is a great basis to basically have different plugins and processes integrated with a viewer. So people have been using NAPARI to build viewers for all sorts of plugins for all sorts of purposes, and this has been a very successful project for that. And then I thought, okay, let's make a plugin that basically adds this LLM, this ChatGPT, Claude, Gemini intelligence into NAPARI to be able to have a conversation with the machine and have it control NAPARI and do things with it. And this is what you're seeing in the background here. You're basically seeing me typing in a web browser and talking to Omega and having Omega do things with the NAPARI viewer. So here's a concrete example. You have NAPARI, the viewer, on the left, and you have a web browser on the right. And you can basically talk to Omega, write to it, and ask things like, for example, change the way we're viewing the data, change the orientation, go to 3D viewing, zoom in, zoom out. You can ask almost anything that you can do with the viewer, either in terms of control, but beyond that, you can also do things like analysis of data. For example, you can ask it, segment the cell nuclei present in the selector layer, and it will answer, I'm using the cell nuclei segmentation tool. So it has access to some tools that let it do things on the images. And so here now you have, in an Apache viewer, you have two layers, think, Illustrator, one layer with the actual image and another layer overlaid on top of that where the segmentation is present. And you can basically see that it has essentially produced a segmentation of the data where each object has a different, label. And you can continue this. Then you can ask, for an analysis on top of that. You can say, please count the number of segmented cell nuclei. And then it's going to be writing code behind the scenes, which you can see here. It writes code that actually in Python that can actually do this analysis. And the result is it counted all the cells there that it has segmented, and there are 317. You can also ask crazy things that at the time, when the first time I tried it worked, but I didn't know it would work. And I had goosebumps when it did it because it's something that goes beyond what normal programs should be doing. Like write a CSV file on the desktop that lists all segmented nuclei together with the area, and then open it in the computer. So because Omega, like any LLM, can't write any kind of code, it can write code that actually, in theory, accesses anything. It could, it has as much access as you have. Luckily, it's a very reasonable AI. Doesn't do crazy things. But if you're afraid of AI going loose, that's oftentimes not happening. I mean, it's not very reassuring to say oftentimes, but basically, I've used these things, for a long time now, and they've never done anything bad to my computer. So, not to say that it cannot happen, but some people tend to, sandbox things to be safe. But in my experience, it's never been a problem unless you ask for deleting all files, and you met, oh no, all the files that I just talked about. Like perhaps the key here is to make sure you're actually very very explicit and very clear about what you're asking. Omeka can see, so you can actually have two images loaded in Apache, for example, and you can ask segment the biological structures on the two loaded images. For each image, please look at them and decide which better tool between StarList and CellPose is the best for the task. So here, basically, it looked at both images, describes them with text. These are not the kind of images that the LLMs are used to look at, but it still makes a good attempt at understanding what it is there. And then because of the tools that it has access to, it can actually do a matching between what's the best tool for the image based on what the tools are, what it knows, what the tools are good at. And so it kind of figures out that, for this image where the cytoplasm is labeled, it's better to use CellPose. And for the image where the nuclei only are labeled, it's better to use StarList because of the convexity of the nuclei. And so basically it runs code for one, it then runs code for the other. And I mean, all of this thing is kind of almost automated, like it's part of this agentic, way of doing things now, where basically you talk to the machine and it has initiative and acts in the world, quote unquote. And here the world is the Apache viewer and the computer to some extent, and does multiple steps that are logical. And so these things can reason. And recently, so I hear a lot of, dismissing of AI in the way of, oh, these are just statistical models that just predict, the next word statistics, and they're not really thinking. I'm increasingly of the opinion that this is actually a flawed way of thinking about it. Our brains are just made of atoms. How could they possibly think? It's just atoms. Atoms don't think. How can an assembly of atoms be thinking? And this is the same thing here. These models are trained in a way that is apparently statistical, and just predicting the next word seems to be a very trivial thing to do. But to be able to predict the next word accurately in all circumstances, you have to have a complete understanding of all the text that precedes. If you have a joke and the last word is the point of the joke, if you do not understand the joke, you cannot predict the last word, which is the the reveal of the joke. So So these models actually, and this is a much longer conversation, but these models actually think. They're emulating thought. They're not as good as we are at this new thing that machines now can do, but they will eventually, if they haven't done that so already, they will attain our capabilities and they will surpass them. It's not a question of if, it's a question of when. And this statistical argument or this dismissal is not valid, especially in light of what's happening right now, what's possible. So one thing with Omega that I find really exciting is you can make widgets, which is perhaps one of the most useful things with Omega, and you'll see with the new thing that Ilan has built. So what you can do is you can essentially add these little widgets that are little interface elements that you can add to the interface of the Parry. So you can make a widget that takes a labels layer and returns a labels layer, but filtered. Only labels with a provided range of areas are kept. And so so you do this, the code is written for the widget, the widget appears here, and then you can start playing with it. You can change the settings of that widget, and it basically starts, generates a new layer here that is filtered labels layer. So now we filter these objects based on their area. So basically anything you can describe and that you can describe with precision, and or that you can anything you can design through a conversation can then become a little piece of code or a longer piece of code that becomes essentially a tool integrated in Napari. So it's like instant plugins for Napari that you can do today and forget about tomorrow, and tomorrow you need something slightly different. So you can design these things completely on demand. You can also just do it directly, like directly affecting, as I showed before, you can directly do things with images. You don't have to create a tool to do things to images, but if you want to use that a little bit, you can use widgets for that purpose. Another widget, which is my favorite, if not the favorite, please write a widget that color projects a 3D stack along the z-axis. The hue of the projected pixel is proportional to the depth of the voxel max intensity. The luminance is proportional to that max intensity, and the saturation is proportional to the contrast between the max intensity and the average intensity. So this is essentially, doing color projection of a 3D stack into a 2D image. And it's not an easy one. I mean, if I give this to a student, perhaps not all students would first understand what I described and then be able to write the code for it, let alone to do it in less than a minute. But Omega does it. Now I have a little widget here, and now I can color project a stack, and the color that you see here, the hue of the color, he encodes the depth of where is that high intensity present in the 3D stack. And this basically is something that can happen like this. You just describe it, you have it, and then you can use it. And actually I've used this. Of course, in the team, most of the people coding are good at coding, so they tend to, we tend to use directly, things like Cloud Code and Cursor and other tools like this to code. But when you're in the context of Napari and you want just to quickly do an analysis or something, or quickly process images, this is actually super practical. Another example is, and what this was actually done very early on, so some of the interface is a bit older. I wanted to double check on the time. Okay, we still have 4 minutes. Okay, so this is a small plugin that just adds essentially adds a little skill bar to the image. So so it first makes a widget that then, well, what happened? Oh yeah, sorry. I think I went a little fast on the video. Okay, so it made the, okay, sorry, I think this video is a little slower than the other ones, and so so this with the waiting, it's writing the code. See, this is kind of old. This is kind of one of the first videos I made. And it makes a scale bar, but the scale bar is black. And so that's not really working. So so this is an example where you can give feedback. You say, look, your scale bar sucks. It's black. And it's because it put an intensity of 1 and didn't realize an intensity in 1 on an image which has maybe 255 levels of intensity. That's going to be black and not white. So I provide that feedback and then makes a new one with a little bit, also some additional parameters that were kind of missing. Well, in this case, the intensity. And now we can have a scale bar where, which is visible and of a given length in microns, and then you have a setting here for the relationship between pixels and microns and the intensity. So yeah. Actually, something I don't have here is that in my slides is that part of Omega, there's also kind of a little editor, code editor, which keeps all the plugins, all the widgets that you generate, and you can then get them back, and modify them and do a lot of things with them. So that's very practical to keep things handy in the Pari. So how does all this work? I see I only have 2 minutes. So I will speed up a little bit. This was a little the joke. I'm not a really funny person, so I try to be funny sometimes, but that doesn't always work. So really, how that works? So it's pretty complicated. There's a lot of moving parts. There's of course a web browser where you talk to the thing. There's the Napari browser itself. We have this. You see here on the. That's my cursor. So this is the viewer, you have the chat window, you have a code editor, which I don't have a demo for, but you can essentially keep all of these little snippets of codes that are generated. You can even send them across the network, like a bit like a kind of, if you have a classroom full of Napari instances running on people's laptops, and they all have Omega running, they can exchange code snippets. There's a whole machinery of how we deal with Python repair of so if the code is broken, it's repaired. Also, we have exception handling. We have the web server, we have the actual agent, we have connections to the LLMs, connections between the LLMs and also the code editor. The agent itself has a loop where you interact with it. There's a memory. And all of this you have to engineer actually. At the time, now it's a little bit easier, but at the time you had to engineer all of that. You just had the completion interface where basically you just provide text and it just gives you the text that comes after that. But the whole agentic workflow had to be engineered. Yeah, so that's basically how this works. Now we are at the half an hour, so I think it's time for Ilan to present his demo.